actually beginning the first steps in a gene expression study. And this is the initial trials looking at a crab that's common in oyster reefs in the Gulf of Mexico called Eurypanopius depressus. And it lives down kind of in and among the oysters themselves and feeds on little worms and algae and other things that are amongst the oysters. We're interested in understanding the mechanisms at a genetic level that allow organisms like this crab to tolerate exposure to oil and to other pollutants. And the way we're doing that is we're going to be experimentally exposing organisms to oil from the Macondo Reserve and to oil plus dispersion to see how gene expression in those organisms change. The first step in the process is to find out how suitable these organisms are by leaving them in the lab for 10 days to 2 weeks, see if they're surviving okay, make sure there's no evidence of stress, and that's what we're doing right now. This is really kind of a lab acclimatization period to ensure that things are suitable. And so this is actually the control, and you see we have a little bit of oxygen bubbling away in there, not much. And these are sealed, and the reason they're sealed is because when we actually do the oil exposures, you have to use a sealed container because oil has a lot of volatile organics in it, and if you don't have a sealed container, all the volatiles will just be off-gas. You have to replace it all the time, or use a sealed container. And so that's actually one of the things we're doing here, and that's in its very early stages. We did some earlier tests looking at hermit crabs and gene expression, and it's amazing the difference in different organisms. We were looking at a common grass shrimp, and we had a lot of deaths in the first 48 hours. Hermit crabs, they didn't seem to care at all. And so it's really fascinating how different organisms handle these types of stresses in different ways. So what we're looking at now is a stereomassus. This is a lobster that's found in deeper waters. This one was collected at almost 800 meters in the Gulf of Mexico. And one of the things you'll see, let me get some of these extra random appendages out of the way. One of the things when you start looking at them is they're rather dirty anyway. They live on the bottom, they live in amongst the mud and the muck. But one of the things that we're finding is an increase in the number of lesions. You can see here the very beginning of lesions, these darker dots. They're actually little sores where bacteria are beginning to eat through the carapace. They actually digest the carapace. And it's thought that this may be in fact tied to the bacteria that are breaking down the oil. And so that following the Deepwater Horizon event, or any other event like that, you would have an increase in the bacteria that are breaking down oil, and when that oil starts to dissipate, you've got lots and lots of bacteria looking for something to eat. And so we've actually seen an increase in the number of lesions per organism, but really in older, larger specimens. So some of the specimens that would be of reproductive age typically are going to have an increased number of lesions. And the other thing that we're finding is gill fouling. And you can actually see, I can take it out if you want, but you see the dark black there on the gills. That is where there is a manganese deposit filling up the gill cavity. And the gills themselves are actually completely covered in a layer of manganese. And while we see both the lesions and the manganese before the spill, it's at much lower numbers. And it's not clear what's causing it. It's not clear if it's actually related to the spill. It's not clear what the mechanisms are. But it's something that we're actually investigating to see if we can determine, okay, are the lesions tied to an increase in the bacteria following that are breaking down the oil? Are we finding an increase in the gill fouling, and is the manganese a result of either drilling mud maybe? Is it a result of something that's in the oil itself? And so So when you look at it, this is again glyphocrangon. Found, let's see, this one was found. This is 1,100 meters. And you can see in this one how underneath the carapace here, the gill chamber, the gills will be found underneath this piece of the carapace. It's very dark, it's a black, and that again is a manganese coating that's all of the gill chambers and the gill bailers. And really, it's unclear how that affects respiration. And when you look at a healthy non-fouled, you can see the difference in terms of coloration. Very distinctive difference. And if we were to open these up and actually look at the gills, so that would be one that we would consider to be heavily fouled. And when you look at a healthy one, so there are a number of offshore hard banks in the Gulf of Mexico that are basically on the outer edge of the shelf, and those banks tend to have a very unique fauna, something that's basically only found on those banks and not in the areas around them. And when we start to look at that fauna, we realize that it has to move across these areas in which it isn't found. And we want to understand is, are these individual hard banks acting as little isolated habitats to which populations are restricted, or do they act as stepping stones and actually promotes connectivity throughout the Gulf of Mexico? And obviously this would have implications for any type of environmental event, any type of event that would affect these different habitats. If you have a population that's restricted to a single seamount or a single hard bank, and that hard bank is affected, you might lose the genetic diversity that's present there.
Simon Pecknick. I'm a graduate student here in Dr. Felder's lab. I just started my master's project, and we're looking at the potential function of a subsurface patch of pigment cells on the antennae of this group of ghost shrimp. And we're investigating the potential function of it, whether it be sensory or anything else. We don't actually know at this point, but those are the preliminary steps we're taking to try and find out what could it be. So it's pretty interesting. It's exciting to be part of something where you really don't know what the results are going to be. But these guys are a group of shrimp. They're burrowers. They live in the intertidal zone, and they're really important for turning over sediments and oxygenating subtidal environments that in many other cases would be anoxic. And it gives the oxygenation of those areas allows for other organisms to come in and inhabit those areas that otherwise wouldn't. So they're a really important species in the ecosystem.
Those little crabs, that's what I work with. They call pea crabs, and they are in a family that is called Pinnotherids. And since they're that small, people have problems like trying to, I'm going to put it here, trying to distinguish the different species. So we're using genetics to see if we can distinguish the different species with using genetic markers. And we actually are finding some species. I'm in the final stages of my research, and I already have like most of the genetic information I need. And now I'm going back and look at them again to see if I find some differences that can kind of support my genetic data.
We're using a genome-wide approach called RADseq that actually looks at tens of thousands of individual sites in the genome to assess relations. And we're looking at a species called Munida angulata. It's a very small squat lobster, and it's probably the most prevalent species that are found in these offshore hardbank habitats.
the evolutionary relationships between different hermit crabs. And I can do that using certain genes and comparing the genes to see how similar they are and how different they are. And then we can kind of get a family tree of the different species of hermit crabs. Well, hopefully right now I'm trying to get a good idea of what species and families are present so that we can look at how the oil spill has affected what species are present, and what species were missing, or what was there before the spill and what's there after the spill. So I think that's one application. So this machine, what it does is it takes the reaction through a series of heating and cooling. And so it'll start really hot, and that really hot temperature will melt the strands of DNA apart. And then it cools off, and that cooling off enables a specific snippet of DNA to anchor on, and that's where the amplification will start. So that's where the enzyme will start, and it'll act like a zipper to create the second strand of DNA on one of the first ones. And it does that at this medium temperature. The enzyme will start to make more DNA to match the first strand. And then it just goes through that cycle multiple times until we have a lot of the gene that we started with, because we need more of it so that we can see it on the gel, and so that later we can put it on this other machine that tells us what the sequence of bases are. So like AGCT, what order they're in, because that's what we compare when we're looking at the different animals, is the order of the A's, G's, C's, and T's. In there is the actual DNA we're going to look at when we go to sequence it in these tiny little tubes. And we're going to look at it on the gel and we can see if it's there or not. So basically we'll see if our reaction worked, which it maybe didn't. But we'll see. Amplification of genes I don't want. So we'll spin it just to get all the water down in there. Sheet of gel has a special dye in it that attaches to the DNA, and that dye glows under UV light. So it'll attach to the DNA, and that way when we look at it under UV light, the places where the DNA, the sections of DNA will glow, and that's how we know if it's there or not, if the DNA that we amplified in that machine is there. So it's got like these little pockets in it, and in each pocket I'm going to put one of my DNA samples, and then we're going to put electrical current through it, and that electrical current is going to move the DNA from one end of the jello to the other, and then we'll take a picture under the UV light in our rocket ship, and we'll get to see the special dye that's in the gel attaches to the DNA, and then that dye glows under the UV light. So every band that you're seeing here is a piece of DNA that has come out of that reaction we did earlier. But that's kind of what it looks like when you're actually looking at the DNA that you have extracted and then amplified.